Thank you, Chris. And I want to just start, of course, by thanking the organizers for the invitation to present in this really excellent forum. I'm going to talk about a topic that I've been spending a lot of time on just recently that's kind of interesting to me. It's partly interesting because I actually spend a lot of my time working on modern, very big data sets and think about big data a lot. And this talk, I think you'll see, is an instance where there essentially is really, really big data from 100 years ago. Finding this data set and sort of exploring how much information was available was one of the really exciting things about doing it. I'm not going to talk so much about that because I'll focus more on the topic of the talk, which is climatic and evolutionary drivers of phase shifts in the plague epidemics of colonial India. And I wanted to address this topic in this forum because we're talking about human-coupled interactions. And in this instance, we have a system which involves a lot of interactions with other organisms, and it involves climatic interactions and as well the complexity of evolution ongoing during the plague epidemics. So I'm going to go through this, and I've got a couple of different objectives that I'm going to go through. First, I'd like to introduce the three major plague epidemics. You're probably all familiar with them, but I want to make sure everyone's on the same page and understands the human consequences of plague over the past 2 centuries. Then I'm going to talk about the particular set of data that I was using, which was gathered by the India Plague Commission. And I'm going to start right out by saying right now, the Plague Commission found some really astonishing and important results 100 years ago in India. First, they proposed an explanation for the seasonality of plague outbreaks. And second, they proposed a reason for the natural decline of seasonal outbreaks that happened over time whenever the plague outbreaks happened in particular cities. I'll explain the details of that as we go on. And then I'm going to talk about the modeling that we've been performing to examine bubonic plague. And what it's allowed us to do is to quantitatively evaluate these proposed reasons for the seasonality and for the natural decline in seasonal plague outbreaks. It's also allowed us to explain unexpected phase shifts observed in Bombay and in Kolkata, seasonal outbreaks, and lastly provides consistency with an outbreak that begins out of phase in Belgaum. So consistent with some of the talks that we had yesterday talking about the kind of predictability of models, what you want models to be able to do is actually demonstrate phenomena across different data sets. And one of the nice things about the India plague example is that there are multiple data sets of multiple outbreaks that occurred in distinct cities over distinct time periods. So we can apply the model in one place and another and see if we get consistency across all of those with the data, rather than just relying on a single instance in a single population. So that's what our agenda is. And I just want to start again by reviewing some of the astonishing events that have happened in human history. So the very first plague outbreak of the 3 that have essentially provided some of the greatest social upheaval in human history was the Justinian plague, which began in 541 AD. And over the next 200 years, plague killed over 25 million people, which in 541 AD was a very large number of people. This is just a map of the Justinian Empire, and there have been claims, although it's historically very hard to know these things, that this plague was one of the components that led to the decline of the Roman Empire. Now, interestingly, in recent years, the Justinian, some of the mass graves from that time, in 541 AD, have been unearthed. This is one particular one in which they found victims of the Justinian plague essentially in building a Munich housing development, and discovered that they could take samples from teeth, and in the modern world, extract DNA from blood that was within those teeth, and reconstruct the genome of the Yersinia pestis infective that is in there. And in doing so, they demonstrated that 541 plague was caused by Yersinia pestis. So some speculations that it had been some other plague historically are pretty clearly ruled out by this kind of data in multiple samples across mass graves. The second pandemic is probably the best known of any pandemics ever, partially because of the outstanding name of the Black Death. Called Black Death, of course, because of the gangrenous the gangrene that occurs in reaction to Yersinia pestis. It originated in China in 1333 and spread along the great trade routes to Constantinople and then Europe. In fact, in the Justinian Empire, it was also new trade routes that brought the plague in. So you can see a very clear interaction between what's happening in human society and the spread of these diseases. It claimed an estimated 60% of the European population. Different estimates range in exactly what they claim, but it was really an astonishing blow to European civilization at the time. Victims of the plague were buried in mass graves, just as happened in the Justinian plague. This is a 19th century engraving, just displaying that kind of burial process. And the mass graves from the Black Death also have been unearthed, and individual DNA sequences from samples also were able to reconstruct Yersinia pestis, establishing that is the pathogen responsible for the Black Death. Now, what's astonishing to me in some cases is that people. So the good side of these things is that people actually were doctors and tried to treat people in the case where more than half of the population is dying over a long period of time. So this is just a diagram of how the plague doctors would outfit themselves. And I just want to point out the stick here, which was used to keep the plague victims away during the treatment process. So there's sort of good and bad sides of this. The really bad side of this, though, is that in addition to the people who were trying to help out in terms of mitigating not just the outbreak, but also helping individuals who had plague to have food and water despite their condition, plague was also viewed by many places at the time as an excellent method of warfare. So a number of strategies have been devised to use plague as basically a terrorism agent from way back when, and it's probably the most commonly invoked method of conducting biological warfare. The techniques have ranged from catapulting corpses over city walls to actually aerosolizing Yersinia pestis or spreading fleas within materials that are being sent to different nations and invoking plague. Of course, these are terrible ways to attempt to do bioterrorism, especially since they're completely uncontrollable and there's no knowledge as to who will actually eventually suffer from these kinds of events. But because of that, it is still marked as one of the Class A bioterrorism elements. So there's some serious concern as to whether one should be able to address plague epidemics in the future. Just to give you the last of the three major pandemics occurred starting in the late 1800s and in the early 1900s, again associated with trade routes. Again, it's believed to originate in China, and you can see that it's spread across the globe in various locations here. And I'm going to talk about one of these locations in this last epidemic in the analysis I'm going to talk about today. I'll just comment, though, that in 1995, the first multi-drug resistant isolate of Yersinia pestis was identified, and essentially what is keeping plague from being a very, very dangerous outbreak. There's two things. One is just the cleanliness of cities. So in modern developing nations, you tend not to have rats in close proximity to humans, and you tend not to have fleas going from rats to humans. That's only in one portion of our human population, though. There's very large numbers of people living in poverty for which this is not necessarily the case. And the other thing that prevents plague outbreaks from growing is the rapid use of antibiotics, to which multi-drug resistance would be a severe problem. And this particular sequence confers resistance to all the major antibiotics that are usually given as protection against plague. So there's some importance to understanding plague. I just want to give you a very simple, I'm just going to talk about one portion of the plague life cycle, which is the portion that's most important for the India Plague Commission results that I'm going to examine. So the bubonic plague pandemics, they're thought to have been dominated by this urban cycle of coupled interactions between rats, fleas, and seasonal weather. So the idea, and I'll give you lots of data on this in just a moment that was gathered by the India Plague Commission, is that you have fleas that are infective, carrying the Yersinia pestis bacterium, It infects rodents. Those infective fleas eventually give the rodent plague. The rodent dies. The infective fleas that are several generations removed from the original fleas then forage for a new host, find it, and spread the disease in this way. Humans are just the unfortunate dead end, so to speak, of this transmission cycle. Generally speaking, unless it's pneumonic plague, which I'll just mention briefly in a moment, the transmission is simply from infective fleas. And there's a very interesting dynamic here, which I'll talk about, which is that they tend to forage mostly for their host of preference, but then if they cannot find a host of preference, then they forage and can find humans. So there are two other cycles I'm not going to go into detail about. There is a sylvatic cycle, which keeps plague around, and that's ongoing. For instance, in the western US, there's sylvatic cycles, and every year or so, someone gets plague. And very often, it's not diagnosed quick enough, and the people die. It has fairly flu-like symptoms, and it kills you very quickly, so it can be a serious problem. There's also a pneumonic plague, which is a little bit more scary because if you get pneumonia from plague, then your cough actually aerosolizes the Yersinia pestis and can transmit it to other people. It's not so common that you get that plague pneumonia before you get to a position where you're not going to spread it, but it does, again, lead to more fears of bioterrorism methods of using this pathogen. So just to go back to these locations, I'm going to talk about India here. You can see the dates here: Bombay, 1896, and Calcutta, 1895. Those are two of the cities I'm going to discuss in my talk today. So again, just going back to the outline, I'm going to talk about the modeling of this bubonic plague, quantitatively evaluate proposed reasons for seasonality and natural decline, and talk about some phase shift observations, which are particularly interesting because although it's fairly easy to put in some sort of cosine function to match seasonality, it's usually very hard to create a model that will actually match phase shifts in a disease situation. And we've managed to do that. So I just want to start by mentioning that there has been some good research on plague. Most of it has asked a very interesting but very different question, which is how the metapopulation of plague remains stable within this sylvatic environment, or how plague arises again and again. It has not so much dealt with the actual plague epidemics and the seasonality of the plague epidemics in particular. So modeling, as I said, can quantify the human outcomes of these coupled interactions. And I'm going to this is my model slide. So please forgive me as I go through the model. It's a necessity in this kind of talk. And right here you see the rat, which is sort of central to the entire model description. This is a susceptible, exposed, and infected category. This is exposed incubating. All of these individuals will go on to being infective. And the point is, infective is actually a category where the rats they die, and then the fleas actually move on to spread to other hosts. I'm not going to go through the equations specifically, but I'll just mention that we do have a carrying capacity term here, and that there's a death term that's regular death, of course, death due to the exposed and infected exposure to plague as well. The fleas obviously play a central role here too. They are being produced. So this isn't a transmission. This is the dotted line indicates an influence. So fleas are produced at a certain rate depending upon the rate of death of rats. Every time a rat dies, they're produced, and the rate. This is one of the things that was measured by the India Plague Commission, amazingly. And those fleas themselves go on to die, and they do it in a way that actually is really important to the story I'm going to tell you. And I'm not going to explain it right now, but there's this term psi of t that I will explain in detail for how they die over time. It turns out to be very important. And then the other thing is, as I mentioned earlier, in previous modeling and in our model, we incorporate differential rates of foraging for rats, which are the preferred host of plague, and for humans, which are the other possible host of plague. So the rate at which the fleas find susceptible rats and cause them to be exposed depends on the amount of rats available. And when rats start dying in a plague, that's actually when humans are infected. And that's when this rate here increases. So it's all conveyed by this mathematical relationship here. So if you infect population, then they become infected and in many cases die. All of this is easily parameterizable. I just want to note that the population in this case is not a typical compartment. We actually have census data for what the population was, and so we just made sure to fit that census data, including the fact that some of these people were dying in our particular because of plague. So that's not a compartment as much as data from the census. As I said, the colonial administration of India was astounding in the amount of data that they recorded, and I'll show you some of that data in a moment. So this basically represents all of our model, except for there's one more piece missing, which is the evolution of resistance within host rats, which is an astonishing thing. But these rats that I talked about are sensitive rats, and typically in a naive population, rats are quite sensitive to plague and will succumb to it very easily. However, repeatedly over the history of time, whenever plague comes out, resistant rats evolve. So it seems to be a fairly straightforward process. And so we're going to monitor that as well, and I'll talk more about the resistant rats as we go on. So let me just return again to the history a little bit. So typical urban tenement looked like this in the beginning of the 20th century in India. This is Dina's chawl. A chawl is a tenement having 10 to 20 rooms in it where people lived. And this is in Bombay. These tenements, of course, had fairly minimal cleanliness standards, and so there were a lot of rats in Bombay, and this turned out to be a major issue in the plague. So this is Jaigali Street in Bombay, had many rats, was stricken with many human cases, just to give you a picture of what the living conditions were there. And these are the markings that the India Plague Commission made in investigating an infested gully on Ali Uttar Street, marking down the kinds of rats they saw and the amount of plague that was observed in those particular places. And this is a typical map of Bombay City with the plague cases and rat infestation differentially marked. So the red is a plague case in this particular case, and they actually went out and detected where they could find rats and just looked at the frequency of rats in different places and then looked at the frequency of plague cases and made the association, of course, between the rat presence and the occurrence of plague. The thing that's amazing is they went on to do experiments. They built these go-downs, which I'll show you one in just a moment, which were used to validate the plague transmission cycle in India. In particular, they had these separated units where they could put fleas, rats, whatever they wanted to in whatever combination and assess whether or not the transmission of plague was going on. And so they validated these the fact that the transmission was occurring by fleas. It's possible for a rat, for instance, to eat another rat and get plague. But when they would do these experiments, they'd put 15 rats in, half of them with plague, half of them not, no fleas. All the rats with plague would die, and none of the other rats would die, and very rarely was a rat eaten by the other rats, even when they didn't provide any food. So they showed fairly conclusively that the transmission occurs via this route. If you can do it by an experimental means, they pretty much did it. The India Plague Commission also conducted massive rat collection efforts in Bombay to try to understand what was going on, and they had all of the colonial power to do this. And you can see here the many collectors coming with the cages that they bring, and then in the back you see one autopsy table. This is just an amazing display of effort here. What they did was autopsy all of these rats to figure out whether or not they had plague or not, and managed to collect these enormous data sets. So this is human plague mortality, and this is the demonstration that human plague mortality followed rat prevalence in Bombay. So I don't want to ask you to read these numbers, but the general point is they're recording deaths from plague, and they're recording the percentage above and below mean for the year. So this is getting to the seasonality question. Here we have the number of rats found infected, and here we have the percentages above and below the mean for the year. And what you see for rats infected, plague-infected rats, what you see is the numbers of plague-infected rats increase dramatically, and then subsequently to that increase as the plague-infected rats come up, as they die, you start seeing what is inferred to be the foraging of fleas on humans, the predation of humans by fleas, and the transmission of Yersinia pestis, leading to this emergence of a following set of plague cases in humans. So this is the seasonality that we're going to try to explain using the model. But I need to tell you a little bit more about where we got some of our data for the model first. So one of the things that the experiments was also done was to validate the hypothesis that flea survival was ambient temperature dependent. So this seasonality, we need to know how it's coming about. And one of the things they did was they decided that probably the best driver for why the seasonality was occurring was the death of fleas differentially based on temperature. And so what they did was they set up flea enclosures, and those flea enclosures were kept at different temperatures by various clever means in the early 19th century, and they assessed the death rate. And so each of these is an individual experiment with many fleas looking at the death rate of those populations. And what they only did it at 3 temperatures, but the regression, by the way, is ours, not theirs. But what we see here is a regression of the death rate against temperature on a log-log scale, and what you can see is that they die at a much faster rate at high temperatures. And so that corresponds with why you have this late outbreak cycle of seasonality in late stages. So those experiments demonstrated this psi of t that we're using in our model, and we used the actual data from the Plague Commission to parameterize the death rate over time for the fleas. We do it in a way that I'm not going to be able to go through the detail in this talk, but essentially what we do is we tie that death rate directly to the seasonality, so that essentially it just provides a slope for how that seasonality occurs, rather than saying that the experiments were exactly how fleas die in reality. But it means that basically the seasonality is constrained to operate according to that slope of death for fleas over time. So these experiments were also conducted to validate the hypothesis that plague resistance increased in frequency in rat populations that were exposed to plague. So in other words, what they showed was that those rats that had been experiencing, those populations that had been experiencing plague evolved resistance. They didn't term it that way, actually, even though they were quite aware of what they were saying. So this is a table of an enormous amount of data showing the percentage of mortality among wild adult moose rattus from various places. And so over on the, my pointer is dying here, but on the, going down, you see different dates, different doses that they gave to the different kinds of rats. And on the x-axis, you don't need to see these names, but are the names of very many different cities from which they collected rats. And in every case, they evaluate somewhere in some experiment on this table what percentage of those rats die when you infect them with a certain dose of plague. And the amazing thing is that in Madras, where there was no plague, you get nearly 100% death. And in the other populations, it varies. But if you, there isn't actually a data set to say like which of these suffered more or less, but it's known Madras did not have a plague outbreak. And the ones that did have plague outbreaks, such as Bombay and Calcutta, have much greater survival to plague in response to plague. So apparently there's a very complex dynamic going on here with evolution, climate, all affecting this epidemic. So experiments were conducted to validate this hypothesis, and this is just another table of similar data, but it's a little clearer for you guys, so maybe you can actually see something, especially if I blow up a part. I'll just show you this part in particular. I hope you can read this, but this is just the percentages dead of plague overall. In Madras, 99%, and then in various other places that suffered much more plague, lower. In Nagpur, which suffered very little plague, 97%, and in Bombay, where there was a terrible plague epidemic for many years, 21% mortality. In other words, 80% of them are surviving compared to 100% after a collection from Bombay. And then they also bred these Bombay rats in go-downs exposed to plague and showed that you could get the resistance level up to 96%. So quite extraordinary ability to evolve resistance over fairly short timescales. So that evolved resistance is really important to have in the model. It's never been included in a model of plague. So if we put it in there, the amazing thing is what it explains. So it explains the seasonality of the plague outbreaks, and I've explained, and the natural decline of seasonal outbreaks. If you have the resistance, that allows you to have a natural decline. But furthermore, it also explains some phase shifts and inconsistencies which we had. So I'll go on and show you that data. So to evaluate the natural decline in seasonal outbreaks in Bombay, here are stochastic simulations we made of that differential equation model. The stochastic simulations allow us to come up with kind of a 95% confidence interval, what we would expect given the model. And what you see down here is again, I don't really have any power on my laser pointer, but we see the actual data. That's in red. The red line is the actual data, and the blue are the outcomes of stochastic simulations matching the seasonal outbreaks. Again, this is the seasonality here is all being driven by that flea death parameter. We force it to a seasonal cosine function, but the actual driver for that is all by the flea death. This connects very interestingly to yesterday's point, which was made, which was that fairly weak seasonal driving. I think it was David Fisman who pointed this out, right? This fairly weak seasonal driving can drive an epidemic. So small factors like when fleas die can matter. We see this remarkable seasonal epidemic. What I show you above is the inferred evolution of resistance in rats compared to the susceptible rat population. It's evolving seasonally because we have a cost. I didn't mention that, but there has to be a cost, or else you can't explain why rats wouldn't just remain resistant to plague over time. Interestingly, that cost, I'm not going to have time to say this anywhere else, so let me just go back on it. That cost, we estimated that cost for every single one of these outbreaks that we looked at, and we got the same cost from all of them. And you can also we also fit the entire thing using the cost that if we make the whole thing operate with one cost, all the fits work beautifully as well. So there's a lot of consistency when you analyze this data. So this is the Bombay data I just showed you. I just wanted to mention that out of that you also get a sort of a regression on the flea death rates. That first plot I showed you was just a regression of flea death rates. There was no connection to the model. When we put it in the model, we get essentially the same. This is a 95% confidence interval from the model on what the flea death rates were using this data, so it's not surprising, but basically there's no inconsistency between the epidemic progression and what the death rates of the fleas actually said, because otherwise this would diverge to a different regression. So the stochastic simulations don't always match everything, but they match pretty well. So this is over 1897 to 1911. The data that we have from the India Plague Commission, of course, was aggregated over months. So what we did was multinomial subsampling to ascertain when the peak possibly could have been, because we don't really know the day of the peak. But in the stochastic simulations, we know the day of the peak. So what this is the day of the peak as assessed by our simulations in blue and as assessed by multinomially sampling from the actual plague cases aggregated from the India Plague Commission data. And you can see they overlap in most cases with few exceptions. Of course, I'm sure there were some things that we didn't model that are responsible for those exceptions. All we're taking into account in terms of climate is temperature. Humidity must have also made a difference, but they didn't keep track of that in the early 1900s. Although they have great temperature records, which allowed us to do this entire thing. So we evaluated the natural decline in seasonal breaks in Kolkata as well. Again, this was after doing everything else. We just ran it on Kolkata, and it fits very beautifully. You see the susceptible rats and the resistant rats evolving in Kolkata as well. By the way, there are reasons for the differential rates at which the resistance might arise, which is that probably the resistance arises as much from rats moving from one city to another as from de novo evolution of anything. And so when you get the later outbreaks, which occurred at later time, Kolkata started later, you get the progression of rats into the population earlier, so then they can spread faster. So here's Belgaum. This is an interesting one because this is an exception. Kolkata and Bombay, or Calcutta and Mumbai as they're now known, are very large cities even back at that time. Belgaum was a moderately sized little, sort of a town. I mean, you can see it's not this multi-story complex city. It had a reasonable population, and it was right near Bombay, so they did in fact get a plague epidemic. The interesting thing about Belgaum is that it happened, and this is just the map cases in Belgaum. Not much detail to say there, but they did all of the research there as well to find it. It's thought that part of the reason, so usually these small places, smaller, more rural places did not get as much of a plague epidemic. There was a significant one in Belgaum, and it's thought it was because in Belgaum they had a tradition of storing their grain sacks inside their own houses, so rats were running rampant inside the houses generally. So if we look at the actual outcome in Belgaum, again, you see a very nice fit, although the population sizes are much different from the large cities in India. Again, you see a spread of resistant rats against against susceptibles. And what's amazing is you see this natural decline in the peaks. That's explained by the resistant rats. But the really cool thing is that the Belgaum epidemic started out of phase, and you can see the very first outbreak had a peak in January. You can sort of see that in the last diagram. Oops, if I can go back to it. That's Kolkata. It's hard to see the phase in these diagrams, but you can see if you look back to the far left here, this is a much shorter duration between the first peak and the second peak. And then what we did here is just, again, plot this looking across the months of the year and the year in which the epidemic occurred. And the data shows that the peak in Belgaum occurred in January, February. It's very easy to fit that point because that's the starting point of the epidemic. But to fit the rest of this dynamic is really astonishing. That ends up fit fairly well. I've showed you the proposed explanations and proposed reasons that were set up by the India Plague Commission. I've shown you that we can quantitatively evaluate all of that really great data that was gathered and never modeled, and actually validate that those are good explanations for why we see the seasonality and why we see a natural decline of seasonal outbreaks. I also showed you that a model of that explains unexpected phase shifts observed in Bombay and Calcutta and provides consistency with an outbreak that began out of phase in Belgaum. So that increase of frequency of resistant rats also yields predictions that are consistent with data from, this is just an offhand sort of little analysis we did. There has been an outbreak in Madagascar in recent times. You can run the same analysis on Madagascar and it fits the data fairly well. We see only the rise of resistant rats very late in the epidemic, but it can still fit that. And I would be a little skeptical of this because it just seems like they came out of nowhere and solved the problem, except that people have done the phylogenetic analysis, and it turns out that you can figure out that the northern rats were back there. This outbreak happened, the northern rats came in, took over the population entirely. The rats from the north, which were already resistant because there was endemic plague then, went into Maharanja and took over that population. Right now, all those rats are all you find there. So there really was a takeover of resistant rats from susceptible rats. So that's the diagram there. And with that note, I think I'll finish. Yeah, so I've shown you that. And just remind you of what I showed you. I've talked about it several times. I want to acknowledge Joseph Lunard, who's the student who worked with me on this research. Very, very brilliant guy. One of these guys who sort of goes off in 10 directions and you have to keep him on the straight and narrow. But other than that, fantastic work he's been doing. I want to thank Dame Sackler and the Sackler Symposium Organizing Committee for the opportunity to present this research.